Right. Hi, everybody. My name is Eli. I'm a Group 2 leader. Some of you know me from yesterday. And we're running a little behind, so I'll try to make this quick. But I'd also like to encourage any questions if they come up during the talk. And so what I'm going to talk to you today about is barcoding, cell barcoding, a way that we can take multiple samples, combine them in a single tube, and run them all at once on either a fluorescent flow cytometer like the LSR2 that we used yesterday, or the CyTOF. So first I'll do a quick introduction to mass cytometry, which I'll probably go through quickly because we've already seen it. Then I'll talk about mass tag cell barcoding for the CyTOF, and a project that we did with this to do some kinase inhibitor profiling. This is a published work, and then finally I'll talk about some current things that we're working on. So first, mass cytometry. You've probably seen this all already, so I'll go through it quickly. We use metal ions instead of fluorophores. We use these lanthanide and heavy metal, rare earth metals, but they're not really that rare. They're non-radioactive. They're non-biological. And we have 100 isotopes available to use, although currently we're using about 40 or 45. This is how we put them on antibodies. You guys have probably already seen this slide, so I'll skip through it. This is how the CyTOF works. Skip through this as well. And go right to the main portion of the talk, the mass tag cell barcoding. So this work is published, and I worked on this project with a former postdoc in the lab, Bernd Bodemiller, and Rachel Fink, who is still in the lab. And we were really, when we first got the CyTOF, we were all very excited to be able to measure lots of new parameters, but we were a little sad because we didn't have a way to do cell barcoding, which was worked out so nicely by Peter Kritzik for fluorescence flow cytometry. And so this work is also published. You can multiplex for fluorescence flow cytometry. In some ways, it works better than on the CyTOF. But if you're interested, this is the only slide I have for fluorescence barcoding. But I would direct you to this reference. It's printed out in the handouts. So the way it works is we take 3 samples, or however many samples we're interested in, and we give them all, or normally we would carry them all through individually through our staining and measurement protocol. With fluorescent cell barcoding, what we do is we label the 3 samples with unique codes, in this case, different levels of this green dye. We combine them into a single tube, add the antibody to do the stain, analyze the sample all at once, and then deconvolute afterwards. So computationally, we can take these 3 populations, separate them, and then look at the 3 populations. Big advantages to this are uniform antibody staining across samples. They're all stained in the same tube, so they see exactly the same antibody concentration. We also have reduced antibody consumption, which is nice, and reduced acquisition time. So when we started using the CyTOF, we wanted to be able to do this for mass cytometry as well. And we took a very similar strategy. Instead of using fluorophores that are connected to cell reactive groups, we use metal chelators that are connected to cell reactive groups. And here we have the DOTA maleimide group, so it can hold on to any of the metals that we use for detection by mass cytometry, and non-specifically label cells by reacting with the free thiol groups. So using these reagents, we can add in any metal, any of the 3+ metals that we want to the chelator. And so we can use as many barcode channels as we want, pretty much, up to 40, I guess. And we use them like this. We use them in a binary manner. So if we have 2 different reagents that we're using to label cells, we can get 4 unique populations that we can then deconvolute after. And in order to multiplex a full 96-well plate, we only had to use 7 metals. So with 7 metals, there are 2 to the 7th or 128 possible combinations. So we use this scheme to multiplex and run samples in 96-well format. Of course, only using 7 out of our 40 available channels left us many additional molecules, surface, and signaling that we can measure. They're listed here. So the study was set up like this. We measured 1 inhibitor per 96-well plate for many different kinase inhibitors. And on each 96-well plate, we did a dose response, an 8-point dose response, and 12 different stimulation conditions. We then multiplexed these into a single well for measurement. And so in each well, we have all of these cell types that we can either come to by manual gating or spate analysis, as I'll talk about later. And then for each cell type, we have this 8-point dose response. So here I'm showing the staurosporin dose response curves. Staurosporin is nice because it's a very broad, non-specific inhibitor, so it gives nice curves for pretty much all of the kinase phosphorylation sites that we measured. So from a single tube, we're able to get 18,000 unique cell population quantified measurements. And that gives us over 2,000 dose response from a single tube. And that's, so for each inhibitor, we have 2,000 quantified dose response. And we calculated IC50s, percent inhibition. So this is a diagram of the signaling pathways that we measured. We wanted to, for this kinase inhibitor profiling project, we wanted to cast as wide a net as we could to get a broad picture of how the cells were responding to all these different kinase inhibitors. This is in the notes, so you can look at it and reference it throughout the talk if you're interested. In order to visualize the large-scale data that we generated, we used this simplified signaling diagram. And so that diagram is now in one of each replicated throughout this figure. And what we're looking at here is 2 things. This first column shows where the cells are activated. So if we have large red circles, that tells us that these signaling molecules are activated by these stimuli. So here we're looking at all these different stimuli and all the different inhibitors. And this is for a single cell type that we manually gated, CD4 positive T cells. And the first column shows activation, and the rest shows the response to each inhibitor. So some inhibitors, like staurosporin, you see inhibit many pathways, even pathways that are not activated by the stimuli. We can have inhibition that goes below basal levels. We can also look at the data not for a single cell type, but for a single stimulus. So here, with interferon alpha, we see mainly or only activation of the STATs. And when we look at all the different inhibitors, we see many different inhibition profiles. Also, we can look at a single inhibitor with all the different stimuli and cell types. So there's lots of different ways to look at this data, and it's quite a large data set. Another thing that's nice, but kind of hidden, is if you go to the materials and methods of our paper, we have a link to our cytobank report. So you can follow this, go to our paper, go to figure 4, A through B, and then if you click on this link, this will take you to a web applet that will allow you to look at every single dose response curve that we collected. And in the top right here, you can pivot by cell type, stimulation type, phosphorylation type, or inhibitor type, and look at whatever curve you're interested in. I was hoping to have a live demo, but we're running short on time anyway. So yeah, let's go over this really quick. So we have IC50 curves, and we needed to determine what was real inhibition versus just noise in the data. And so we used the R squared for how well the sigmoid was fit, as well as the fold change. We also compared the data we collected to in vitro kinase inhibitor profiling. And the agreement, perhaps surprisingly, was good. We're measuring two different things here. We're not looking at in vitro single kinase inhibition. We're looking at the cell's response and downstream effects. So you would think that maybe there would be some differences and we do see some differences, but there's a strong correlation between our results and these in vitro studies. So, go quickly through SPADE. Aaron will be talking about this after the break. So, this is our SPADE analysis for the data that we collected in the paper. So we measured 10 cell surface markers, these 9 plus CD45. And using these 10 markers, we were able to identify multiple cell types. One thing that reviewers were very interested in when we got our comments back from the manuscript was comparing multiple patient samples. So we did that. We compared 8 unique PBMC samples, and we found that, at least in terms of surface marker expression, they were pretty similar. Maybe, I don't know if that's surprising or not. It was maybe a little surprising to me. And so here we see CD3 expression, CD33 expression, and also, importantly, the signaling response was quite similar between the patients. Here we have S6 compared to unstimulated control, and also SET5. If we look at all the signaling responses together, and we compare all the patients to every other patient, we see a very high correlation. Another thing that we did was look at the time course. So for all of the previous slides, the time point we were looking at was 30 minutes. We were looking at a single 30-minute time point after stimulation. But we were also interested in the temporal response. So instead of testing dose response for a single drug, we did no drug with 8 different time points. And showing that here, there were lots of interesting things that came out of this. One thing that was interesting was with the LPS stimulation, we see a NF-kappa B phosphorylation at 15 minutes. In the this is in the monocyte branch. So monocytes are relatively early responders to LPS stimulation. And then later we see STAT3 phosphorylation in T cells and BTK phosphorylation in the B cells. I quickly want to mention, so this data set is public now, but there's still a lot of interesting analysis that can be done. So one thing that we found very interesting was, are these cells talking to these other cells? Are they secreting maybe some kind of cytokine? Probably. Or are these cells responding just to the LPS, or are they responding to the monocytes responding to the LPS? So some experiments that we've been doing more recently are asking this kind of question. And I'll go through this quickly. Basically, working with Manfred Claassen, we can look at the derived network from our data set and see how it changes over time and in different cell types. We can look at signaling response of different cell types and look for correlations in time. And then once we come up with models and hypotheses, we can test these. And this is still with Manfred Claassen. So he did lots of depletions, because we were interested, if we get rid of the monocytes, will the other cells still respond the same way to these different stimuli? So here we immunodepleted CD33 positive cells. And if we compare immunodepleted to mock-depleted samples, we see that after stimulation, multiple cell types, the blue is the depleted, so we see a reduced response to multiple stimuli in many different cell types. So the monocytes seem to be, I mean, for those of you who know more about immune response than I, maybe this is maybe not so surprising, but we think that the idea to find these relationships de novo is very powerful, and we're continuing to follow up on this. So finally, we've been working on improving our mass tag reagents and methods. So one thing that we've been very interested in is coming up with new reagents that use new metals to expand the number of antibodies that we can use to study whatever we want to study. And we've also come up with methods for arranging the cellular barcode to remove doublets from our data. We can talk more about that later. If you're interested, come and talk to me about that. So, in conclusion, mass cytometry is very useful for us. We are very fond of mass tag cell barcoding. This work is published with our kinase inhibitor profiles. And of course, the SPADE algorithm is also very useful, and Aaron will be talking about that later. And that's it. I'd like to especially thank Bernd and Rachel for the work that I presented, for helping me and working. It's been very fun working with them on the work that I presented today. And everybody else in red contributed to the kinase inhibitor profiling project. And thanks for your attention. I'd be happy to take any questions if anybody has any. Yes?
For your 24 plates, were all combined into one?
Yes. Actually not. I probably went through that too quickly. So each plate of 96 samples was combined into a single tube, but we didn't combine the 24 plates together.
So,
yeah. Yes.
How many cells did you have to collect to get enough, and how many sample injections did you have to do to get that?
That's a great question, and very relevant for those of you who are working with mass cytometers. So we collected, on the average, between 1.5 and 2 million cells per 96 well plate. And what really saved us, or made it possible, I guess you could say, is that all of our cell types that we collected were identical. This is the same PBMC sample, just with a different stimulation. So if we had tried to do this with different cell types, or maybe if like cancer samples or things where each well had its own unique populations, it would have been much more difficult because the cell numbers would have become limiting. But because we could take all 96 samples, pool them together for the cell type gating, that made it workable. And so you asked how many injections. So usually, I don't know, I guess probably around 30 injections. So 2 million cells. So we're at the site off for maybe 2.5 hours. Per plate, yes.
Can you
Yes.
then use less cells per well, so then you would need if you just collect them separately? Or
I think it depends on the question you're asking. So if every cell type is the same and they're just differing in how they're stimulated, as the case for us, then I would say yes. But yeah, I think it's really application specific.
So would it be an advance if you just have few cells per whatever, so that you can just use less cells to make a whole bunch of stimulation conditions? Would you still need the same amount of cells as you just would narrow it per cell alone?
You may be able to get away with fewer cells. It's always better to have more, I guess, is the easy answer. But one thing that is nice, we noticed that when we have fewer cells in the tube and we do a lot of washes, we tend to lose a higher percentage of them. So the fact that early in the washing process, we're combining all the samples together to give us a large number of cells in the tube and a large, or well-defined pellet does reduce cell loss compared to running all the samples individually.
So Eli, I'm still
Yes.
a little confused on how many cells you actually ran. You're saying you had 1.5 to 2 million cells per well
No, sorry.
combined?
So after combination, so we put about 15 million cells onto the whole plate. So I guess approximately 150,000 cells per well. And we combined them and then ran them on the cytoff, and we would run and collect about 2 million cells total from the multiplex 96 well plate.
Okay, so the 1.5 to 2 million are the actual events.
Yes.
The cell events
Yes.
that you collected.
Exactly, yes.
Because there is a huge loss of
Yes.
cells running through. So are you getting 15 or 30% recovery of cells that you actually inject?
That is the average. We frequently, we don't count the cells before we inject them. We usually just stain them, intercalate them, and inject them without counting. If we did count them, we could have an accurate number for you, but we didn't. But I think that a 20 to 30% recovery rate is normal, so that's what I expect. All right, great. Thanks, everybody.